Well, welcome everyone to our closing keynote speaker for our Love Data Week this year. We have with us Melody Baddoo. She is a native Louisianan who earned a Bachelor of Science in Biochemistry from Xavier University of Louisiana, and then a Master of Science in Biochemistry and Molecular Biology from LSU Health Sciences Center in Shreveport. She began her career at Tulane in 2000 in microbiology at the Tulane National Primate Research Center. In 2002, she moved to the Center for Gene Therapy, where she studied mouse mesenchymal stem cells. Close, excellent. I was a biology major before I became a different kind of doctor. In 2005, Melody joined Eric Flemington's lab in the Tulane Cancer Center to manage the proteomics COBRA core. When the NGS Bioinformatics core was established in 2010, she transitioned into bioinformatics, specializing in processing and interpreting next-generation sequencing, or NGS, data. So she is currently the manager of the NGS Bioinformatics core, where she supports researchers across the Tulane Cancer Center and broader Tulane community, LSU, and the University of Florida in processing high-throughput sequencing experiments. Melody's interests center on data literacy, reproducible analysis, and helping investigators build confidence in meaningful engagement with their data. Her talk today is titled From Black Boxes to Biological Insight, Teaching Researchers to Truly Engage with Their Data, and explores how researchers learn to engage with data, drawing on real-world experiences from a bioinformatics core to examine confidence, pedagogy, and the design choices that shape data understanding. So please join me in welcoming Melody.
Thank you so much, Lisa. Thank you very much for that introduction. And so I'm going to begin doing a couple of things. First, I'm going to thank KP for the invitation, and I appreciate it so much. Before I get into my talk, I have this slide up here just so I can tell you a little bit about the bioinformatics core, a little bit more than what Lisa shared. So we're at the LCRC downtown, 1700 Tulane Avenue, next to St. Joseph's Church. We're on the 8th floor. And it's a room maybe a little smaller than this, fairly nondescript. There are a bunch of computers in my offices across the hall. But we've just been providing the hardware, the software, and the manpower to help investigators keep their hands on their data. And I'll talk a little more about that in the talk. But as of June of last year, we are now on iLabs. So anybody who wants help can access our service on iLabs. And the link to our iLabs site is on our website, waveseek.tulane.edu. This is the site I built. It's fairly unfussy and gives the facts. And reads really nicely on cell phones now. The first site I built was kind of raggedy, and this one's much nicer. So you can find more information about the core and about our resources, our publications, etc. Waveseek.tulane.edu. That's where all the information lives, and we're at the LCRC downtown. All right. Okay. Let's do this. All right. So I love talking about data. Oh my God, let me calm down. All right, I love talking about data, all things bioinformatics data especially. I love it. It's a treat to be able to stand in front of you and do this. This is a talk that I give like all the time, pretty much. I've never ordered it so nicely as I hope that I have for you today. But anytime there's a student or a postdoc or an investigator who comes in the core and they say, Melody, I found this data. I need you to help me download it. I need you to help me process it. I need you to tell me what it means. And when I'm ready to publish, I need you to write the methods for processing, okay? I give basically this talk when they come in the core and just kind of, hold hands and walk them through this. So it's a treat to be able to share this with you. What I observe and the way investigators engage with their data, how they learn from it, and what I learn from them. So I love to do this. When I said yes, yes, I'll do it. And when I was asked for a title, this is the one I gave. And so the first part from black boxes to biological insight. It makes sense. It reflects what I observe, the way that investigators engage with their data. They start with, well, what is this? Well, how do I open this? Well, what does this mean? To, oh, snap. This is a lovely list of differentially expressed genes. And wow, these are some, I didn't realize that those splicing alterations were going on, in my particular experiment. So it goes from really, I don't know what I'm doing, to goodness, this is, thank you. What else can we do? And when we're ready to publish, we'd love to have you as an author, to which I say, oh, thank you. Make sure you spell my name right. 2 D's, 2 O's. The title, the first part describes what I observe, the way that investigators engage with their data. And the second part, it speaks to an aspect of my job, teaching people. I mentioned early in the first slide that we're on iLabs. So we're you pay a fee and I'll perform a service for you. So there are investigators who don't wish to learn about this stuff. They don't want to spend the time. Here's my data, process it for me, please. How much will it cost? And I'll process it from tip to tail. When I say tip to tail, I mean from raw digital data to a very nice report with figures and quality control and graphs and Excel spreadsheets and all of the things. I was like, this is a pretty good title. That'll do. But then I started building the talk, and I was like, oh, snap. This is the one that I wish I had submitted. Because I think, same talk, this is a better title. Pedagogy, design, and learning from inside the core. And again, not just the learning that I witness from like students and investigators, but what I learned too. I was like, oh, this is a better title. All right, moving on. So before I get too deep in it, I want to tell you, which I think I just said, like I could give this talk without slides. I could give this talk just standing up here doing this. I don't even need a whiteboard. It would be helpful for some of the figures and stuff like that. But my husband says that I probably should be a cartoon because I'm so animated. But yeah, I could probably give this talk without any slides, and I could talk for 2 hours straight too, like barely taking a breath. But I'm not going to do that because I'm going to respect your time. And so I have these slides to keep me on track, all right, to respect the time I've been given. To keep me focused. Like I have something I want to share with you. I want to tell you something about the core. I want you to learn something about the core and about bioinformatics and NGS and engaging with data. And so since I have something I want you to learn about that, these slides provide structure. And structure helps learning, right? Okay. So there you are. And I'll say also too, like me sharing with you that I had one title and I changed another. To another, I realized that there was a better one. Me sharing that I could give this talk without any slides. Like that's all very relevant to what you'll hear later on. And so I hope I convince you of that. Speaking of learning, I didn't learn the word pedagogy till a year ago. Never even heard the word before, never seen it before. So I love talking about data. The only thing I love talking about more than data is my husband and children. So I am mom to a couple of teenagers who are at Willow right up the street. And my eldest is a cellist and he is graduating high school this year. So excited. So this time last year, we started doing the things, right? We started touring colleges. And so he's a cellist. He's interested in pursuing music in college, to which his dad said, what? And I was like, hey, that's his gift. We're here to foster it. All right, but I digress. So we're getting ready for these college tours. And so because mom is, checking lists and, doing the things, I was reading the bios of like the music faculty, all right? And I would see in their bios, my research interests include performance and music theory and pedagogy. And then, I flip and I said, what is that? Okay, whatever. You read a word, you see it, you keep it moving. And then I would, look further down, and I was looking specifically like at the strings instructors, and I would see, so-and-so, adjunct professor of cello pedagogy beneath. And I was like, what? Is that some music specific word? Like what? I'd never seen it before. And so I was like, well, what does it mean? So I did what I tell my children to do, and they were like, Mom, I'm like, you have a computer in your hand. Look it up. So I did that. I looked it up, and Google says that pedagogy is the method and practice of teaching, especially as an academic subject or theoretical concept. I said, oh, okay. So very simply, pedagogy is the strategy and methods used to help students learn, all right? It's adaptive, that's important too. It considers the learner's needs and their background, all right? I said, well, shoot. Pedagogy is not some music-specific word. Pedagogy is just how people learn. And the strategies used, right, like I said, to teach them. It's not what they learn, it's how. I said, wow. So when I was extending the invitation to do this talk, I see that word again, and I realized that I've been applying a pedagogical framework. See, it's a new word to me, okay? I've been applying a pedagogical framework in the way that I train and teach and share with investigators in the core. Like I've been doing that all along. Didn't even know it, that was the thing. I've been doing that in the way that I train them to use different tools to handle data, to process data, to manage data, to move data, to organize it. I've been doing all of those things. And I didn't initially think of it that way. So Lisa shared with you that my undergraduate and graduate degrees are in biochemistry and molecular biology. And so I was a lab rat. That's like my little handle on, little thing is lab rat. And in 2010, when my supervisor, who studies EBV, this guy's always been like cutting edge. I'm so blessed and so fortunate just to be surrounded by really smart people who say, you can do that and you can handle it and here's a task and I'm sure you can do it and figure it out and I'm here to help you. Anyway, so in 2010, my boss had employed RNA-seq to investigate gene expression in latent and lytic EBV cultures. And he says, Melody, we're going to start a high-throughput sequence analysis core. Do you want to be involved in that? I was like, okay. And that's how it started. So prior to 2010, my background was I don't have a degree in computer science or computational biology. So this is all what I've learned and am continuing to learn and build upon since 2010. So pretty much self-taught. So like I didn't think of what I am doing, what I'm doing in the core as, pedagogy. I thought of it like I learned how to do something. I'm going to teach you, I'm going to share with you how I learned to do that thing. That's it. That's what I thought it was. But now that I've learned the word pedagogy, I'm thinking that I should probably add that word to my LinkedIn bio, pedagogy. So I'm thinking about it. All right, so before I dive into, pedagogy and design and learning from, to engage with data in the core, I'm gonna tell you let me take a couple of slides. I'm gonna tell you about NGS for folks who don't know. So NGS stands for Next Generation Sequencing. It's also referred to as high throughput sequencing. And it's just an umbrella term for different sequencing technologies. RNA-seq, ATAC-seq, ChIP-seq, Hi-C-seq, RIP-seq, CLIP-seq, CAIDS-seq. There's a bunch of seqs. It's faster and cheaper than Sanger sequencing, and it has allowed for greater advancements like in molecular biology and genomics and oncology. I work in the cancer center. So the NGS workflow can be broken down into 4 steps. 3 of which I'm going to share with you right quick. So that first step is extraction, all right? So when nucleic acids are extracted from either cell culture or tumors, they can be fresh, it can be samples that are formalin fixed, paraffin embedded. You can isolate the nucleic acids from those tissues. And there's a multi-million-dollar industry for extracting nucleic acids from all kinds of samples for high-throughput sequencing. The second step is library preparation. Now, I'm not going to hang out long here. Basically, is when that genetic material when those nucleic acids are prepared for sequencing. They're fragmented, adapters are added, and they're ready to go on for the next step, step 3, which is, this is just a little graphic just to show a sequencer onto the platform. Samples are actually loaded. The device is called a flow cell that goes into that sequencer. And it looks like it's about the size of a microscope slide, and it's got like 8 lanes in it. And the libraries are actually loaded onto that lane, onto the different lanes in that on the flow cell, and then that's onto the sequencer. And basically, they're amplified and sequenced to determine the order of nucleotides. Okay? Okay, all right. So it's at that last step, analysis, where the bioinformatics part comes in, all right? The output here from sequencing is what's called a FASTQ file. That's your raw digital data, okay? So prior to, and like people still use it now, so I don't wanna talk bad about microarrays, but prior to bioinformatics, the technology for looking at gene expression was microarrays. And that can be considered analog, where high-throughput sequencing is, you can consider that digital. Well, it is, you get digital data, all right? So this last step is bioinformatics, where you get these FASTQ files. A FASTQ file is just a text file. I'll talk a little bit more about it later on. It's just a text file. For every sequenced reads, there's 4 lines of text for every read. FASTQ files are usually delivered compressed, and they can be on the order of a few hundred megabytes to several hundred gigabytes in size. All right, so this stuff is hard to wield. If any decent RNA-seq, let's say from somebody's studying a breast cancer, and they have MCF7 cells. MCF7 is a breast cancer cell line. And they have MCF7 cells in culture, and they take 2 samples, 2 different culture, slow down. They take 2 dishes of MCF7 cells. One they treat with something, the other they don't. They harvest the genetic material, have those sequenced. Any decent RNA-seq experiment where you have like an N of 3, so 3 replicates for each condition. Minimally, there's going to be maybe about 50 gigabytes of input compressed. Okay, so uncompressed, it could be 1.5 to 2 times more than that. All right, so I want to impress upon you the size of this stuff, and you're not going to be opening this up in Excel or TextEdit trying to work with it on your laptop with your 8 gigabytes of RAM. It's not going to happen. So yes, on to bioinformatics. So I told you a little bit about next generation sequencing, and this graphic, I just want to show you that it lists like the components of bioinformatics. It's computing. You need some hardware. You need some software to be able to process this data. It's managing the data. I just shared with you how large these samples are, and that's the raw data. So before processing, what's cool about bioinformatics and processing high-throughput sequencing data is that the size of your data start up here. With your raw data. As you move down with processing the data gets smaller and smaller, all right? And then towards the end of the processing, you don't have like a bunch of fancy files that you need fancy programs to open. You can open them with, TextEdit or Notepad or Excel. I wouldn't advise you do so, but you can, okay? So managing the data is important. I spent a lot of time collaborating and communicating with investigators to try to figure out their needs, reading papers, going through databases to see what publicly available data that we can download and process for whatever questions they're trying to ask. Down here at the bottom, it says problem solving. It probably should say under that too, firemen, because I spent a lot of time putting out fires when things happen, and then developing tools to process high throughput sequence data. So again, bioinformatics combines all these skills to go from what is this to biological insights. Bioinformatics helps us give meaning to the data. I mentioned we started this in 2010. We started the core in 2010. So here we are 16 years later. And now next generation sequencing is a widely used technology among a lot of researchers. But believe me when I tell you that there are still significant bottlenecks that exist, even though the technology's been around for a while and everybody's doing some sequencing. Seek this, whatever. So the bottlenecks remain for investigators who aren't computer scientists, who don't know how to use the sophisticated programs, who don't know how to handle the data, who don't know how to manage the data. And so that's where I come in. I mentioned that we're service for a fee. Not only will I process your data, but we want to help investigators keep their hands on their data. When I first started doing this, I'll pause for a second. When I first started doing this, and actually there's a gentleman he's uptown. He had some sample sequenced. So he sent the samples off to be sequenced, and they did the sequencing and the bioinformatics processing of the data for him, okay? And he got like this 30-page report back. He didn't know what was going on. He was like, I don't even know what this means. He was like, we paid $3,500 just for them to process the data, not the sequencing. He was like, I don't know what this means. I don't know where to start. I don't know how to tease it out. And I don't know how he heard about us. This was in 2013 because we had just moved to the JBJ. I don't know how he heard about us, but he came over to the core. He was like, yeah, we got this stuff sequenced. I don't know what this means. And I processed that data all over for him. And believe it or not, so it was over the course of a year, we processed the data. And he emailed me like 2 weeks ago. He was like, yeah, I'm ready to publish. I was like, dude, that was 200 years ago. But what's so cool is something that I'm gonna share with you later on. Because of the framework that I had set up. Like I did not know that I was doing this, but because of what I had set up back then, I said, all right, I know we have his external hard drive here with the data. And he sent me an email very clearly with what he wanted. I went to my stash. I have a little file cabinet full of hard drives from back in the day when we had stuff on a bunch of hard drives. And I said, oh, that's it. And was able to connect it to my computer, go right in and go, yeah, that's it. Draft an email to him, and he was like, oh, okay, we'll have you, as an author on the paper. Thank you very much. 2 Ds, 2 Os. Spell my name right. So yeah. Now into the nitty gritty of it. So I talked about that the technology's been around for a while. I talked about that analysis at that portion in the NGS workflow, so extraction, library prep, sequencing, analysis. The analysis is where bioinformatics comes in. I showed you my first title, from black boxes to biological insights. So what's the black box problem? That problem is the situation where you have some input, you put it into something, out comes something, but that middle part is hidden from you. You don't know what happened. So in this instance, the input would be the raw data, the FASTQ files. The system that I would be inputting those FASTQ files would be a tool to align the reads to a reference genome, or if it's some organism where there's no complete reference genome, I might do something called de novo assembly, where I stick all the reads together to assemble a genome, okay? It might be where I run the reads through an aligner to look at long-range chromatin interactions, and you get bed files and bed graph files. So what am I trying to say? That the black box problem, the complexity of all the stuff that lives in the box that's hidden from investigators, and they're not bad, right? We use apps on our phones, all right? The work that I do in the core, we don't use a lot of graphical user interfaces, okay? So apps. We work in terminal, so we enter that stuff, we can use code, Python and R and Java and Perl, Unix commands, to actually run the code that's going on behind that application. All right, so black boxes aren't bad, but what they do is they don't facilitate learning. Okay, unless you design against it. Okay. All right. So what's inside the black box? Is it magic? When I first started training, like students and investigators and stuff like that, I know what the aligners do, and I know what the programs do for differential expression testing and peak calling and format conversions. But I was doing a lot, and had to move very quickly from one thing to the next. And so I would sit down with someone and I said, okay, what do you have? Do you have ChIP-seq? Do you have ATAC-seq? Do you have RNA-seq? Do you have small RNA-seq? And they said, okay, we have this. And this is when I was training, not when I was in instances where I was processing the data for them. And I said, all right, you're gonna take your FASTQ files, we're gonna put it in here. Magic, you get an align file, right? So I was like, it's magic, but it's not. I talked about aligners. Oh, sorry, I'm trying to stay on track, Melody. And how it seemed like a black box to researchers. All they are is tons of different aligners. All they do is they take your raw FASTQ reads and they map them to a reference genome. That's all they do. The different aligners, there are specific aligners depending on your input. And so you need to know. So if someone comes to me and they said, oh, okay, I need to align these reads that were sequenced on PacBio's instrument. All right, well, PacBio generates long reads. And when I say long reads, I mean anywhere from about 500 to 10 KB in size. Okay, typically, the Illumina is the company that's cornered the market on short reads. And those machines are everywhere. And the length of the reads that come off the Illumina platform are anywhere from 50 to 300 bases. All right, so why am I telling you all this? Depending on your input, that determines the type of aligner that you need. All right, and so I said aligners they all do the same thing. They just map the reads to a reference. But what are you asking from your data? Do you just want to do differential expression testing? So you want to see from these MCF7s that are untreated versus these MCF7s that were treated with tamoxifen, what genes are increased, what are decreased, all right? If you just want to see that, well, then you can use this aligner called STAR. It's called Spliced Transcripts Aligned to a Reference. It's memory intensive. So if you trying to run STAR like, on your laptop or one of the desktops we have, it's gonna hog a lot of the memory from the computer. So you're not gonna be able to do too much else. And it's reasonably fast. If you are not interested in, I'm sorry, STAR is best for differential splicing analysis. If you are only interested in differential expression testing, so looking at differences in gene expression from one condition to the next, then the better aligner to use is called Callisto, all right? STAR aligns reads to the entire genome. Callisto is faster than STAR, uses less memory, but it only aligns reads to the transcriptome. So all of the RNA molecules, all right? So it's a smaller, think of the entire genome as a really big pond, right? Okay? And the transcriptome is much smaller that reference set that you're aligning your reads to. So it's much faster. So those are the types of things that I help investigators with when they come to me and they say, I'm trying to do this. So yeah, the aligner one chooses depends on the platform. Where were those reads sequenced? How long are they? What you want to explore. Do you want to explore alternative splicing? Do you want to look solely at differential expression testing? Do you want to identify variants? So where in your samples do the bases differ from the reference genome, indicating some type of mutation? Okay. Yeah, so inside the black box isn't magic. It's all the formulas and the algorithm and the code that's doing the things that you want to do. There. But that's not all that's inside the box. Inside the box is also pedagogy. It's what do you want to do and how do you handle it and the attempts and the decisions you make and interpretation and the questions you ask. That stuff's also in the box. And I'm going to circle errors because the way that I treat errors is a very intentional choice that I made. Again, I didn't even realize I was doing this, y'all. But the way that I treat errors is very intentional in the framework for teaching. Let's see, what did I say? So pedagogy. I said that pedagogy was adaptive, that it depends on the learner's needs and their background, right? So one of the first things I do is, for someone who wants to be trained to process their own data, to keep their hands on it, is I ask them a series of questions. Okay, have you ever worked, with are you a Mac user or a Windows user? Because we're solely, Macs and Linux in the core. So I'm a bit of a PC dummy. I can, stumble across, but under the hood of a Mac is Unix. And so it's already built in, and the computers have a lot of processing power, much more processing power than Windows. So that's why we use those. Okay, all right, moving along. I talked about my pedagogical framework design in the core. So I touched briefly on just now on how I address errors, but I want to back up. Earlier I said I could give this talk. I really don't need slides, but I have the slides because I have something I need to stay on track and I need to respect the time, and I'm doing all right because I need structure, and structure encourages learning, okay? And so the way that I've structured hardware, workflow, pipelines, the way that I teach investigators how to name their files and name their directories. And it might say, Melody, that's not a big deal. Oh, yes, it is. I just gave you an example earlier about how back in 2013, 2014, a gentleman came, and then 2 weeks ago, and I did some work for him, and then 2 weeks ago, he says, yeah, I'm ready to publish. It was because, number one, I kept the drive, but that when I connected the drive and looked at the folders, I know exactly what this is. I didn't have to pull and also I have like a file cabinet full of notebooks too. So I didn't have to pull my notebooks out. Everything I needed was right there, and I could look quickly at the way the directories were named and figure out what was done. So I was thinking about my future self, right? And so that's how I try to, when I train investigators about organization and management best practices. I talked about earlier how when you first start with these FASTQ files, that this data is huge. It's super important for if you're doing any high throughput sequencing, any data, any files that you can have compressed, compress them. It's really, really sloppy to have a bunch of uncompressed FASTQ files or SAM files milling about, taking up space, either on your external hard drive, either on Cypress, Hideki, right, on Box. That's sloppy to have those uncompressed files like that. So those are the types of things about organization and management. I mentioned earlier we work in terminal, so I don't use very many graphical user interfaces where you click a button and browse and drop a file. We don't do that. Everything we're typing in code, or we're running Python scripts or R scripts or things like that, right? So when you're working in terminal does not like blank spaces in the names at all. So if I were going to make a folder on my desktop and I were going to call that in terminal, and I'll call that folder Melody Baddoo February 2026. I would not type Melody space Baddoo space February space 2026. I wouldn't do that in terminal, because terminal would see that as, you want to make 4 different folders. One called Melody, one called Baddoo. You see where I'm going? So those are the types of things. And so I say it now. It makes sense. But someone who's never done it before, who's not in the habit of doing so, y'all, especially like with people who, are brand new and I'm helping them along and something will go wrong. They're like, I ran the command, but nothing happened. And I'll go to the computer, I'll look at the terminal. Or if I'm feeling really lazy, I'll just log in from my desktop in my office. I'm like, this file, the file's not found because you're in the wrong place, or you never made this directory, or that's not the name, or there's a space at the end. And terminal sees that. You don't see it, but there's a space at the end of the name. So it's those types of things that, again, didn't realize what I was doing, that this was pedagogy, but this was how I learned. I told you that my background prior to 2010 was not computational science. I made a lot of mistakes. Spent a lot of time just, oh my gosh, I'm such a dummy and, an imposter, and nobody's gonna believe I know what I'm doing. So that's why I teach the way I do. It's important to think about your future self as far as naming your files. And then too, you might go down a path and this naming structure for these directories, this work. And then 2 weeks later, when you get your next experiment, or when you have to reanalyze the data using a different reference genome or something like that, you go, oh snap, maybe there's another way to do this, right? But I showed you that too, because I submitted one title, I started doing the talk, wait a minute, this one's a better title. So it's dynamic, change it. Take the time and fix it. Your future you will thank you for that. All right. Before I go on. So there's the way that I structure the hardware, workflows, and pipelines and those types of things. But there's also a really intentional design that I employ when I am training people. All right. And so there's the two things I want to, there's a lot. I'm painting with broad strokes because I don't have 10, 15 more minutes. So the first is interpretation. All right. And then the second is addressing errors. I'll talk about interpretation first. So most systems are not designed, I talked about that, to teach you. They're designed for minimal human input, maximum automation, and completion. All right. And so my little graphic here is this kind of the face of an app, a graphical user interface. And then you got some flow diagrams and some things going on in the background. That's behind it. You never see that, right? And then here, what I'm trying to show is that the goal, next step, check your list. The next step, it's not designed to teach you anything, all right? And I'm gonna give you an example of a tool that I use all the time. I can take my glasses off because I can see this. All right, so for quality control, I use a tool called FastQC, all right? And it's usually the first stop when I get raw reads. Doesn't tell me if an experiment is good or bad, just tells if it looks funny and if we need to do any pre-processing before I do the alignment or before I do the de novo assembly or something like that. So and it's a neat program. It runs fast, and what's cool about it is that you can run it as a GUI, as a graphical user interface, as an app, or I have some code that I wrote for processing RNA-seq. So you can also run it, in terminal. So you can, the business ends of it, and you don't have to, have an app, and it generates the same output files. So it's cool. It's a workhorse. I use it all the time. And what you'll see is that your output for FastQC, it generates an interactive HTML file. And what you get is these green, yellow, or red flags, okay? And the green means, of course, pass. All right, we can figure this out. The yellow is warning and the red is fail, okay? But there's no explanation about what any of that means, especially, so green, okay, right? But the warning and the fail, there's very little information you get about what that means. And so this is an example of an RNA-seq, one of the FASTQ files from an RNA-seq. So I invested, my supervisor who studies EBV, we have a PO1 large collaboration with some investigators at the University of Florida, and I'm on the grant. So when I process all their RNA-seq for the people in Florida. Gosh, I'm not gonna go there because if anybody wants to talk about it later on, I'm happy to. All right, so this is an example of one of the FastQC reports from one of the FastQ files. And so here we're at the per base sequence content module, which is right there. That's the first one where we get a warning, all right? And this plot reports the percent of bases called for each of the 4 nucleotides across all the reads, all right? And you have to know at what type of file to look for. So FastQC can process, can do quality control for any FastQ file from any platform, but you gotta know what to look for, all right? You have to know what to look for if those FastQ files are whole genome, so if it's DNA-seq or if it's RNA-seq, all right? And so for RNA-seq, here's the rub. So it says here, we get a warning, all right? And we can probably figure this out. That is, the warning is probably due to this foolishness right here, right? But here's the rub. For any RNA-seq that was generated where the libraries were prepared with the library prep kit, the TrueSeq library prep kit, they use random hexamers at the beginning. So it'll always fail, all the time. But they don't say that. So an investigator will see that and think, oh, do I need to trim the first, 10 to 15 bases? No, that's par for the course. That's what it looks like. So here's a very wonderful app, but it's not designed to teach you anything. It's more designed just to freak people out, all right? Some of those, when, the fails, so what is the per base in content right here? If you see a red or a yellow X right here, like, pay attention to that, all right? So at that module means, so when samples are being amplified and sequenced on a platform, if the platform can't discern that it's an A, C, T, or G, then they'll just put an N there, all right? Ends are, what can I do with that? Nothing. This is garbage. And a lot of the earlier sequencing, like when we first started doing this, a lot of the stuff that was on like databases on ENCODE and stuff like that, it was filled with garbage sequencing, just a bunch of N's. And you have to you got to filter all that. You can't map that to anything. And it takes up space and it's useless. But again, we're in the year of our Lord 2026 and everybody's using high throughput sequencing and the technology's gotten better. And it's very rare now to see sequence with a bunch of N's in it. But if were I to see that for this module, were I to see a warning or a fail, I would make a beeline straight to it and try to figure out what's going on, all right? So that part about it, is informative. But I know about that, right? Okay, because this what I do. So what am I trying to say? Long story longer. The design impact is that the flag shows you the problem, but it doesn't tell you how to fix it. It doesn't tell you anything about it. You don't learn anything, okay? All right. So I told you about what most systems design for, and I gave you an example of a real tool that I use all the time, all right? I talked earlier about interpretation as far as of one of the intentional design choices that I make. And it's important to realize that even though the digital data that comes off your sequencer is basically a text file, you can't treat it like text files like we're used to, right? So if I had a list of all the grades for the G-Chem course, from 2017 to now, right? Yeah, you open it up in Excel and, sort and filter and do whatever, right? But you can't do that with NGS or Bioinformatics text, even though they're text files. You need specialized tools for handling those, all right? And the clue, even though these are all this stuff, these text files, all right? But the clue that you probably shouldn't open in Excel is the extension, right? .fq stands for .fastq. You'll also see .fastq, all right? The .gz means it's compressed binary. I can take a peek at a binary FASTQ file in Terminal, but it looks like gobbledygook, so it's useless. So the key here is when I'm working with a student, when I'm working with an investigator, is telling them, yeah, this is basically a text file, but these are the different tools that you need to process this or to handle it or to take a peek at it, okay? So yeah, part of what I do is teaching people to recognize that this stuff looks different than what they're used to seeing. I'll go back to this slide, just a general reminder, most systems aren't designed for learning. I think I've pretty much covered that, and it's obvious when you think about it. It's not a flaw in apps. It's a plus that we can move quickly and I don't have to write code, right, to log into my bank account. I don't want to do that. I want to use the app, right? So it's not a flaw. It's a plus. But it's just not always conducive to learning. And it's like that makes sense. It's kind of like, it's dark at nighttime, right? Of course it is. But it's, when you're working with one of these systems that isn't optimized for learning and you're met with an error, like it still surprises us. Like, well, what happened, right? So yeah, so what I try to do is overcome that and design around that in the core. And one of the ways I do that is how I address errors. I told y'all earlier when I first started doing this, I made a lot of mistakes. Oh my gosh. And I had to learn how to interpret errors. So say for Excel, and I'm going to talk about Excel in a minute, a computer's pretty stupid. It's only going to do what you tell it to do. If you tell it to look for a file in one place and that file is not there, it's not going to say, I'm sorry, I don't know what you're talking about. And a lot of times a little fun experiment I do, especially when I'm training like graduate students because they're so eager and, and undergrads and stuff like that, I will sit at the computer and I, give my little spiel or whatever and point them to some resources. I said, okay, I'm going to drive and I'm going to step you through this. And then I step back and I said, all right, you drive. All right. I said, I'm gonna tell you something. I said, I'm not trying to be ugly. I said, I don't want you to think I'm being mean. I said, but I might let you mess up. I said, and I'm not trying to be ugly. I said, I want you to get used to seeing that error, not freaking out about it, and knowing what it means, knowing how to ask the next question so you can overcome it. And so no such file directory is common. It's common. You're in the wrong place. You put in the name of one file or you misspell something, computer is not going to say, well, R sometimes does this, which is cool. You can type in something and R will say, did you mean to do this? So I like that about R. But if you're just running like in bash, it's not going to say, did you mean to say, January 2026 instead of January? It's not going to do that. All right. It's just going to say error, file not found. And so what's the computer telling us? You asked me to do something and whatever you ask me to do, I can't do it. All right. So what I try to do is I try to force the next question. And so that's why I sit back and go, all right, I might let you mess up, and they always do. I chuckle every time. But what does that mean? That file not found error. What does that mean typically? It means that you're not in the right place. You thought you were in one folder, but you're not. It means that maybe you ran a command and the file you thought was generated wasn't generated. Or maybe it was generated, but in your command, you told it to go somewhere else, right? Not the right place. So what do? Does the file exist? Are you at the place you intended? Breathe out. It's cool. We'll figure this out, all right? And then I know over the years, I've learned some neat little code to do a find file type. There it is, okay? So it's not mysterious, right? You get comfortable with the error, and know how to deal with it. So I'm not trying to teach you necessarily how to fix it. I'm trying to teach you how to think about it the next time you see it, all right? And this is part of the workflow. This is part of what I do. And what happens when you, like I just said, when you stop seeing this as, oh my God, I messed up, I failed, feeling a little bit defeated. You're like, oh, okay, this is how it is. I can figure this out. I can do this. And then if you can't, they come across the hall. And if I'm feeling like I need some steps, I'll get up and go over and do it. If I'm not, I'll log in from my computer and see what's going on. Yeah. All right. Getting close to the end. I'm wrapping up. So what does this mean in the core? It means what I said earlier, that we're not just a service. You pay me and I'll process your data. But it means you can come and learn, and I can learn. Every time, y'all, every time I get a new project, and it's like being in my favorite class in college. There's this lady from, an investigator from downtown, and she's doing a really, really interesting metagenomics study. So she's investigating changes in the gut microbiome in animals that are preclinical, not preclinical, in normal or wild type animals versus those that have Alzheimer's disease, right? And the last time I did any metagenomics, y'all, was 2014. And so she reached out to me. She said, I got your name from such and such. They said you did some RNA-seq for them and they were very, very happy with it. And they said you could help me with this metagenomics. And I said, yeah, I can do that. I hadn't done any metagenomics since 2014. But I knew tools existed, and I was like, okay, it's just and then I have, 4 2023 Mac Pro computers. I have access to Cypress. I have access to HyperGator, Florida's HPC. I have, 2 Linux computers in my lab, one of which has 2 NVIDIA A6000 GPUs. I can do this. And I had so much fun processing that data for her. And when I gave the first round back to her, she was like, oh my gosh, this is fabulous. Do you want to collaborate? I can put you on a paper. What did I say to her? Spell my name right. 2 D's, 2 O's. So we're a learning environment. And my pedagogical framework, the way I've designed things, all right, that's quiet pedagogy. That's you learning in the way that I'm teaching you how to handle your data, in the clues that I'm giving you, in me teaching you that, when you start down one road as far as organization of your directories, it's okay if you, 2 weeks later or a month later or the next experiment, you go, I need to rename this. And I always encourage you, take that hour or 2 and rename these files, rename these directories and move this stuff around. Or if you don't want to do all that, make some directories. If you want to rename files, right? Make new directories, move the things you want to where you want them to be, and then write a little README in each directory for your future self. README about this folder. What's in this folder? This is data I processed from such and such. This is publicly available stuff that I downloaded. This is where it's located. That business. So, yeah, and support and ownership. What did Eric say when we started the core was we wanted to help investigators keep their hands on their data. And the gentleman I mentioned who's here is a prime example. He farmed the sequencing and the processing out, got the process data back, got his 30-page report, and $3,500 later had to come and have me do it over for him because he didn't know what any of it meant. Yeah, I'm gonna close. I'm going to thank my boss, my supervisor, Eric Flemington. He is boss extraordinaire. And just really, he taught me how to write. Oh my goodness, I thought I knew how to write. He's taught me how to write. And he's a little bit persnickety as he's gotten older. I've been with him since 2005. But really just kind of reinforced in me the importance of the details and thinking about my future self. He attracts really, really wonderful and smart students. So I've had the opportunity to work around some really, really smart graduate students and watch them go on to do great things. Prescott Deininger used to be a director of the Cancer Center. And anytime the people who give money came around for tours, he was always, and this is the core, and this is Melody, and she's doing great things. So he was a really big cheerleader for me, and still is, and I appreciate that very much. I thank Carl Barabalt from the Cypress HPC team, and Hideki, thank you too. These guys have been really, really helpful. When we first started with our first computer and getting our first, program installed, Carl came and spent, I think, all day with us just trying to get it installed, and something now I could probably do in about 5 or 10 minutes. The Tulane Cancer Center and the Louisiana Cancer Research Center members, I work with a lot of great people, a lot of smart people. Makes my job easy. And what they probably don't know is a lot of them I consider really, really great mentors. And I'm grateful for that. I wouldn't be over here today if I never met Stephen Oglesby and KP May. So I appreciate you. Thank you very much. And thank you, Lisa. And thank y'all for y'all time. That's it. Any questions?
So in CAIDS, our focus is really teaching students data literacy, and many times the students that we attract are not usually from STEM fields because there's sort of this perception that STEM researchers as the scientists don't really have any kinds of anxieties or issues or fear of working with data, but it sounds like you run into some of those STEM folks that do. So can you maybe give some examples of some of the data fears or anxieties that you see when the researchers come in asking for your help? What are the things that they struggle with, or the parts of working with their data that they tend to be the most scared of?
I'll give you an example of retrieving the data, and then when it's time to deposit it. There's a lot of anxiety I see at that point. Whether it's publicly available data, or let's say they had their samples sequenced, they might have had them sequenced at the CAIDS lab in the JBJ. And so what the CAIDS lab do is they will upload everything to BaseSpace. So BaseSpace is like Illumina's app for processing and managing data. And so they might upload everything to that BaseSpace and then send an email to the user and said, okay, here are your credentials. You have 30 days to get your data. Well, where am I supposed to put it? And how do I move it? And usually they, when that email comes to them, they typically don't have any idea about the size of it. So they don't know. You don't want to put this on your laptop, right? They don't realize how long it's going to take sometimes to get it. They're unaware of some of the tools that they can use to move this data quickly. I mentioned earlier that I'm part of a PO1 with the University of Florida. So with Florida, when we first started moving things back and forth with them, they had a Globus account, all right? And because I have credentials at University of Florida, I was able to get access to Globus to move data from our external hard drive or our storage straight to their HPC. Fast. And Globus is set up, like Tulane has Globus now, which is great. And I'm trying to introduce that to some users because it's a great tool. But so for example, if I was trying to move some data from my desktop to Box, all right? And if this data is pretty substantial in size and I'm trying to just click and drag it over to Box, it's not the best way to do it. Something happens and it stalls on Box, all it's gonna tell you is failed. That's it. So you get what? Well, why? What happened, right? But if you use Globus, you can start the transfer, and if it fails, it'll pick it back up again at the place where it left off. And then you get detailed information about, okay, this transfer is complete, going to the next. So to answer your question, I see a lot of anxiety when it's time to retrieve that data. If it's data that they generated, meaning they had sequence, or whether it's publicly available data. Some of the databases to retrieve the data, you need some software to do that. It's not just click it and it'll download to your computer. Even from a popular place people get publicly available data now is a European Nucleotide Archive, all right? You can go into the ENA, put in the sample name, so the accession number, and it'll populate. It's like, oh, these are all the samples associated. Even if you say, well, I only want these 5, you click on those 5 and say download. What it'll give you is a script that you would then have to take to terminal to download it. And so a lot of people are kind of scary about, anxious about that. When it's time to submit data, so we've got over that hump. When it's time to submit data, when you're ready to publish, you have to submit your data to a database. And that's no small notion. And I've done many Zoom calls with, can you control our computer? So I'm trying to help them well. The issue is GEO, so the Gene Expression Omnibus is where a lot of data are submitted. There are very specific rules about how they want the data formatted for submission, and the curators on the other end expect a thing. And if you don't do the thing, they will tell you quickly, that's not gonna work, So I see a lot of anxiety there when it's and a lot of stuff in between, but the most when it's time to retrieve data and when it's time to submit it. Thank you. Thank you so much.